affects the resolution of experimental ARDS through IL-4 mediated lung macrophage reprogramming by the primary author D'Alessio, Craig, Agarwal, and the research was conducted in the Division of Pulmonary and Critical Care Medicine at the Johns Hopkins University. So let's start by finding out what ALI, or acute lung injury, is. Acute lung injury is a condition that is characterized by hypoxia or deficiency in the amount of oxygen to peripheral tissues due to an effect to the lung. Acute respiratory distress syndrome displays a severe continuation of acute lung injury. There are many problems that arise as a result of acute respiratory distress syndrome. There are currently many pharmacotherapies and research revolving around reducing the mortality of the disease and morbidity of survivors. And currently, it is a huge clinical problem because there are no specific therapies that exist. Acute respiratory distress syndrome, or ARDS for short, is a lung condition that does not allow proper amounts of oxygen to enter within the lungs. This would then mean that there would be poor circulation of oxygen within the blood. And ARDS is also caused by many indirect and direct injuries, such as aspiration, which is breathing into the lungs, inhaling toxic chemicals, pneumonia, which is an infection that inflames the air sac in one or both of the lungs, and septic shock, which is an infection by bacteria, fungi, viruses that causes organ injury where toxins initiate a full body inflammatory response, such as low blood pressure and abnormalities in cellular metabolism. And finally, trauma. Some of the symptoms that are involved are breathing abnormalities, low blood pressure, organ failure, and rapid breathing. Oftentimes, ARDS leads to an accumulation of fluid in the air sacs or alveoli, which would prevent oxygen to enter within the bloodstream, and the level of oxygen in the blood could result in dangerous levels. Macrophages that are shown to have a resolution to ARDS and ALI. There are connections that show that lung inflammation is connected to monocytes and macrophages. The phenotypes of particular early macrophages involve M1 and M2 macrophages. M1 macrophages usually promote tumor growth and invasiveness. They typically attack tumor cells and present antigens to the immune system. In response to signaling through pattern recognition receptors, macrophages become M1 and they are able to produce levels of pro-inflammatory cytokines, which would basically ensure inflammation to occur. Acute lung injury models show that the macrophage transforms from an M1 phenotype to M2 phenotype during the start of lung repair and restoration of tissue homeostasis. M2 macrophages are thought to have contributions in wound healing, limiting cellular inflammation, cytokine production, and fibroproliferation. These M2 macrophages are typically initiated by IL-4 and IL-13, which are typically cytokines. A cytokine are a small group of proteins that are important for cell signaling. It affects many target cells and has importance in antibody production and inflammation. They are produced by hematopoietic cells such as T cells and mast cells and basophils. They're found to promote airway inflammation by initiating cells such as eosinophils, macrophages, and dendritic cells. It has also been found that IL-4 helps with the proliferation and activation of fibroblasts, and also mucus productions through the stimulation of airway epithelial cells and goblet cells, and also airway hyperresponsiveness through the activation of smooth muscle cells. IL-4 cytokines are also found to be initiating M2 macrophages, as said before, and the paper focuses on whether exogenously administered IL-4 could induce M2 macrophages that would essentially improve lung repair and the resolution of experimental acute lung injury. On the fact that exogenously administered IL-4 could help initiate the production of M2 macrophages, which would then help with the resolution of experimental ALI and enhance lung repair and function. So here are the materials and methods. In this experiment, they utilized male C57BL6J and MALB-CJ mice that were about 8 to 10 weeks old. The IL-4 was prepared to an antibody-IL antibody, and each dose complex contained 2.5 micrograms of recombinant IL-4 cytokine and about 15 micrograms of an anti-IL antibody within a 150 microliter solution of PBS. In order to mimic the effects of ARDS, the researchers utilized intertracheal lipopolysaccharide or Pseudomonas bacteria administration in order to display experimental acute injury, lung injury, and to understand the resolution phase of acute respiratory distress syndrome with the use of IL-4 and M2 macrophages. Analyzed lung histology using hematoxylin and eosine staining. They also did a lung function measurement as well as a whole lung homogeneity RNA and protein isolation, as well as a real-time PCR, a Western blotting, and flow cytometry. So there were four specific results that were found within this research, and it involves, and I'll describe the materials and methods utilized that are appropriate for the results. So the researchers were able to find out there was an improvement in the mortality of the mice, and it would speed up the resolution of experimental lung injury. Using the same treatment, they found that it also reduces fibro proliferation and improves lung mechanics after lung injury. The macrophages found within the mice were found to have also expressed M2 markers after administration of the IL-4, which accelerates ALI resolution. With impaired M2 macrophage activation, the mice are unable to benefit from IL-4 treatment, which is found in one of the figures, and the increase in resolution of experimental lung injury through the administration of IL-4 treatment. With the treatment of the wild-type C57BL6 mice, they placed these mice with a high-dose intratracheal LPS, followed by an IL-4 treatment for several days. So the research sought to understand the IL-4 administration using certain protein complexes. So there was these complexes called matrix metal proteinases, MMPs for short, and they are in charge of lung injury and repair by regulating components such as collagen and elastin that are found within the extracellular matrix. So seen within figure 2C, the research measured the expression of MMP2, MMP9, and MMP12. For MMP9 expression, there was a reduced expression, but for MMP12, the expression was upregulated, which is seen within days 4 and 6 compared to the other groups that were seen. Through this upregulation, they were able to find that IL-4 administration assisted with improvement in lung repair and as well as lung injury. To supplement this, they also found that the IL-4 treatment in LPS exposed mice, there was a recovery level in the H2O groups and the diffusing capacity, which measures physiologic lung recovery and repair. So for figure 4A seen here, the researchers wanted to find out if the IL-4 mediated M2 had a role in behavior of lung macrophages and whether the pathways and mechanisms that were involved would help assist with improvements in acute lung injury and lung repair. So researchers found out that there was an increased expression of M2 markers RG1, FIS1, and YM1, which is seen by the black bars denoted by intratracheal LPS and IL-4 administration. And this was used by using an immunoblot, and it was confirmed by using a Western blot, which is a technique that detects specific proteins in a sample of tissue or extract. It basically uses the principle of gel electrophoresis to find out the importance of impaired M2 macrophage activation and how it correlates with IL-4 treatment. In order to test this, they compared the response of the intratracheal LPS between the wild type mice and the STAT6 mice, which have a deficiency in the transcriptional regulator of M2 differentiation and activation. Within the data, we see that there's an increase in the BAL protein and BAL neutrophils at day 5 for the LPS, IL-4 treated wild type mice. But for the IL-4 treated wild type mice, with STAT6 mice, there was an impaired resolution with elevated BL protein and neutrophils. There was a decrease into marker expression in STAT6, along with a decrease in the expression of MMR and dectin-1, which is seen in the figure. In addition to this, we are able to see in another figure, the expression of CD86 is also decreased, which overall presents the essential use of STAT6 dependent M2 protein regulation that is needed for IL-4 to help with experimental acute lung injury. So overall, we find that through the IL-4 treatment, it improves mortality and speeds up resolution of experimental lung injury. As well as using IL-4 treatment, we found that it reduces viral proliferation, improves lung mechanics after lung injury when identifying these markers called MMPs. We also find that macrophages express M2 markers after the administration of IL-4, which also accelerates ALI resolution. And along with that, we find that with the impaired M2 macrophage activation, they're unable to benefit from this IL-4 treatment. So overall, what's the point of this whole research article? This research article shows future therapeutic studies involved with M2 macrophages and IL-4 treatment. The problem with ARDS is that there's not a lot of clinical trials being done, as well as therapeutic studies that are being evaluated. This can overall benefit in using IL-4 treatment on acute lung injury and progressively through acute respiratory distress syndrome. Thank you for listening to my presentation.